Hi, this is Mr. Adler. Welcome back to Ciencia on Fuego. And today, we're going to be talking about cells. Now, this might seem like a small topic to you, but without cells, you couldn't be alive. Your body is actually made of trillions of cells. The average human adult, their body is composed of 100 trillion cells. If you go and you look at a small part of yourself under the microscope, you'll even see these cells. It's really neat. You can take a toothpick and rub the inside of your cheek and go and put it on a slide and look at it with a microscope. These cells do everything for you. They help you breathe. They help you break down the food you eat. They help fight bacteria inside of you. Your cells are your superheroes. They do all of these incredible jobs that keep you alive. And those cells, well, they're pretty complex, even though they're small. Your cells are actually made of lots of smaller parts called organelles. And one of those organelles is called the cell membrane. And that's actually what we're going to be modeling today. One more time, the organelle we're focusing on is the cell membrane. Now that's the outside of the cell. The cell membrane is kind of like the security guard for the cell. The cell membrane gets to determine what comes in and what comes out. But there's a catch. I've got here a couple of plastic baggies. And to simulate what happens at the cell membrane, I've got two chemicals. I've got some simple iodine that you can just pick up at the pharmacy. You might use it to treat cuts. And I've also got some cornstarch. Now, I'm going to set up two experiments. First, I'm going to put cornstarch in a baggie. Now, this part doesn't need to be really specific or accurate. I'm going to put one big spoonful of the cornstarch inside my baggie. And I'm going to be really careful to only get the cornstarch on the inside. Once I do this, I'm going to add a little bit of water. Now, if I was going to be accurate, I would add about 100 milliliters of water, but I can just get it pretty close. There we go. And I'm going to go ahead, I'm going to mix that around. Now, this is like my cell. The baggie itself is the cell membrane. Inside the baggie, this cornstarch water, that actually models something called cytoplasm. Cytoplasm is the stuff inside a cell. You can think of it like the jello inside of a cell that holds all of the other cell parts, like the nucleus or the mitochondria. And maybe we'll learn more about those later. Everything outside of the baggie in the cell membrane, well, that's the environment outside the cell. Outside of your cells, maybe there are other cells. Maybe there's blood or other bodily fluids. Now, to model what's outside the cell, I'm going to use a cup. And inside this cup, I'm going to add some of my iodine, just a squirt or two, and again, a little bit more water. Now, I'm going to put my cell, my baggie, with my cornstarch and water inside of my iodine, which models the outside environment. But I'm also going to do the opposite. I'm going to take a baggie, and I'm going to put some iodine in here. This time, the iodine water is going to be the cytoplasm inside the cell. And again, I'm trying to be really, really, really careful to not get any contamination. I squeezed the iodine inside the bag, and I made sure none touched the outside. One more time, the baggie, that's the cell membrane. Inside the baggie, that's the cytoplasm. And now I need to make the outside environment, which this time will be my cornstarch. And I'll finish off my water. Take my cell and my cell membrane, Drop it in. Now, this experiment takes a little bit of time, so I'm going to set these off to the side. Now, you might think nothing is going to happen. Plastic bags, they have to hold stuff, so they probably shouldn't have holes inside. And that's right, if I put food in, I wouldn't want things to go through. But if I go ahead and look at one of these experiments that I set up a little while ago, you're going to see something really interesting. Hold on just one second. Now, normally I have my students leave these overnight, and in fact, I prepared these ones a while ago, so you should be able to see a change. When I take the bags out, you'll notice that nice white cornstarch water isn't so white anymore. Starting over here, I got this pretty dark purple color inside. My iodine looks the same. Over here, the same thing happened, but in reverse. Inside of my baggie, I've got this purplish color where that white cornstarch used to be, but my iodine is that same like brownish yellowish color. So what happened? I didn't poke any holes in my Ziploc bag. The difference is only one of the chemicals was able to cross the bag. Starch is actually a huge chemical. It's made of tens and tens of atoms, all bonded together. Iodine is actually only made of two. It's I2, two atoms of iodine bonded together. It's very small. As a result, what happened in both of these experiments was the really small iodine was able to cross through the bag and into the cup of cornstarch. Same thing in reverse over here. The iodine was able to cross through the bag from the cup into the baggie of cornstarch. But because cornstarch is such a big chemical, it wasn't able to cross into the bag and then in the cup. That's why the iodine still appears the same color pretty much as when we started. Now, it's not obvious that those have any holes in them, but this models something really important about the cell membrane, the idea of selective permeability. Yep, that is a big word. I'm going to say it one more time. Selective permeability. You might also have heard it as semi-permeability. Now, let's break down that word. Semi means half, like a semicircle. And permeable means when something can go through something. So for example, if you're playing football and the offensive line keeps letting the defense in to sack the quarterback, you might say, oh gosh, that's a super permeable offensive line. Well, your cell membrane is semi-permeable or selectively permeable. Just like the iodine was able to cross through the plastic bag and get into this baggie of cornstarch, important substances like nutrients and oxygen can cross through the cell membrane and get into your cells. However, things like bacteria and substances that are too large, they're probably not able to cross through the cell membrane to get into the cell. One more time, we just modeled the idea of selective permeability. The cell membrane doesn't stop everything from coming through. It's not a wall. It's more like a security guard at a gate. Some things can go through, nutrients, oxygen, water, but not everything. And that stops you from letting things into your cell that could harm you, or from letting things leave your cell that you want to keep inside. I hope I've sold you a little bit on cells. It's a little topic, but it really does build up over time. See you soon.